Okay. All right, welcome everyone. Welcome on YouTube. We have an inside job today. So I'm going to do the introduction in a moment. Let me start the recording. Here we go. Record to the cloud. All right. We should be good. Megan, give me the thumbs up on the YouTube once we are streaming. Yes, we are live. Okay. All right. Welcome everyone. Thanks for coming out today. So we have a very special homegrown guest here. This is Nicholas Chen. So Nick is a fourth year PhD student in my lab, and actually I was fortunate enough to have Nick in class as an undergraduate in 2017 or something. Yeah, in an ecological engineering course back in the day. Then he got his undergrad at UFES, and then he did his master's at USF in business analytics and data innovation or something. And then so he saw the dark side. He did a little bit of consulting, and then he decided to come back and get his PhD here. So Nick, you have the floor. So cool. And maybe I'll hide this. Any of this clicker? Still, we just do a little. I think probably not. Maybe that was me. Yeah.
Hi guys, so as David said, my name is Nick Chen, and first off, I'd like to thank the contributors and contributing organizations for the support on this work. So today, I want to talk about using self-organizing maps to identify the drivers of Florida red tide dynamics. Okay, so what is red tide and why is it hard to study? So for those of you who have seen the news, I'm sure you've seen about these fish kills and respiratory problems and dead fish on the beach and the millions of dollars of lost revenue for the state every year. So red tide is caused by this dinoflagellate species known as Karenia brevis, and red tide has become this nearly annual occurrence along Florida's Gulf Coast. And so the reason why red tide is so hard to study is because there's this nonlinear relationship to explanatory variables. And basically that means, okay, just because you have more nutrient loading doesn't necessarily mean you're gonna have worse red tide blooms. And so I want to direct your attention to the little plot we have below. And what that was, this index that this group came up with to help quantify red tide. And essentially the darker and taller that bar is, the more widespread and intense the red tide is, right? So another reason why blooms are so hard to study is because they have this highly variable magnitude, frequency, and duration. Sometimes you get these blooms that last like a month, and sometimes you get blooms that last anywhere up to 2 years. Okay, so they actually use this in situ data from the Florida Fish and Wildlife Conservation Commission, and they began sampling in 1953. However, it became a little bit more systematic in 1994. Before then, it was a little bit more event-driven. So those are biased towards more severe events. So the FWC hosts these sampling data with the cell counts and water depth and different data on these different sampling events. Most of the red tide studies use the data set from FWC, these in situ samples, as a response variable in their models. However, it's difficult to still, when you're building these models and trying to figure out what causes these blooms or what's causing the intensification of these blooms, it's difficult to interpret the spatial and temporal extent and dynamics just using the in situ samples alone. So to help support this work, Dr. Ron Fick created these red tide prediction maps that combined remote sensing data and these in situ maps to produce these 1 kilometer daily 1 kilometer resolution red tide maps from 2002 to present day, or 2024. And what they do is they classify pixels, so the 1 kilometer resolution pixels, into 6 classes. So anywhere from 0 to 1,000, basically meaning no bloom, all the way up to 1,000,000 plus cells per liter, or really bad bloom. And so when we compare it to our BSI, like the BSI, we have this magnitude, and you can see the really bad 1,000,000 plus cells per liter in those yellows up in the estuaries and along the coast in these daily 1 kilometer resolution maps. Likewise, you have this duration, and you can, having the daily resolution, you can actually track how that bloom is growing and shrinking over time. And then the events. So we can actually see that event started in like July of 2006 and went through 2006. likewise with the BSI in the extreme to the left. Okay, so how do we actually go about characterizing these blooms? So, there's certain states that we're interested in as scientists, like there's this no bloom state, and there's this initiation state when it kind of gets off the ground, growth, and then eventually this decline and termination of red tide. And looking at like BSI and our maps, we know that all blooms are not created equal, right? These red tide blooms are this coast-wide phenomenon. We see them over various estuaries like Charlotte Harbor down Southwest Florida and Tampa Bay and Sarasota. And these blooms have this offshore and inshore component where we know that Cape brevis typically initiates offshore anywhere from like 10 to 40 miles. And through some confluence of events, you get these estuary inshore bloom events. So these blooms grow and shrink with different intensities over different spatial extents. And it's highly unlikely that every bloom event is caused by the same sequence of events. So how can we use these higher spatial temporal resolution maps to our advantage to understand and characterize these bloom dynamics? Okay, so some of the previous work that's gone into studying red tide have identified that these bloom dynamics are likely the result of, but not necessarily limited to some of the variables you see below, such as ocean circulation patterns. So there was a study that came out a few years ago that linked this retracted position of the loop current, which is this big ocean circulation pattern of the Gulf of Mexico, or whatever it's called now, body of water due west, and red tide events. So the hypothesis was that these anomalous upwelling events would cause the proliferation of this phytoplankton species that ultimately outcompeted red tide. So, and when you had that retracted position, you would have this, lessened or non-existent red tide event during those years. There have been some studies linking nutrient loading and watershed inputs to red tide, showing this like complex lagged effect through phase space diagrams and the intensification of these blooms, likewise, showing nitrogen loading from Lake Okeechobee and this correlation between that and the intensification of these blooms. And finally, there have been a few machine learning studies that have taken all these variables into account alongside other climate conditions like wind and temperature, and showing how the importance of some of these variables to the intensification of these blooms. And a lot of these studies have identified is that there's no individual variable that has this strong linear relationship to bloom events. There's always a lot of asterisks within these papers. And because there's a lot of complex mechanistic combinations of events that are leading to them. So traditional mechanistic ecological models are prone to overparameterization and are data limited. And some of the machine learning methods have produced really promising results, but they're hard to interpret as mechanistic. Okay, so the hypothesis of this work is that bloom dynamics, some of the things we talked about before, like initiation, growth, and termination, are caused by a nonlinear combination of ocean circulation patterns, watershed inputs, and climate conditions. So this is, that kind of summarizes some of the previous work where you were feeding in some of these features of temperature and stream flow and loading and wind and ocean circulation patterns, and trying to map that relationship to red tide concentration. And we've produced like high R squareds and F1 scores, but things like that alongside feature importance are not necessarily causality, right? So the objective of this work is to to take all of the features and responses, in other words, our temperature and watershed conditions and a red tide, and try and cluster them into these environmental regimes. And so the study uses self-organizing maps to identify these reoccurring environmental regimes that correspond to different red tide states. Okay, so let's walk through a little example here, and I'm going to refer to this as like temporal clustering. So this very simplified, example of what we're trying to do here. Let's just take stream flow and temperature as our two variables, and our response being red tide concentration. So again, in the simplified hypothetical example, what we're looking for are different regimes with past data and how that corresponds to a tide state. So in other words, are there states where you have peak stream flow that are low and peak stream flow and low and decreasing temperature? Does that correspond to no bloom states? Likewise, do you have regions where there's high and decreasing stream flow and low temperatures? Does that lead to bloom initiation? And some of the other examples below. The key is to capture enough representative historical information that corresponds to each of these dynamic states. And the reason why we believe we can do this is because environmental variables show a lot of collinearity. There's a lot of them are cyclical in nature, and you see a periodicity to a lot of these. Okay, so in some of our previous work, what we tried to do was identify what ingredients do you need to bake a cookie, right? And we found that tires and clip art fish and pencils really weren't that important. And things like butter, sugar, teaspoon, flour were more important. I forgot eggs, they're expensive. The new question is, there are a lot of ways to make a cookie. We're not a cookie. And what are the quantity of ingredients and the steps required to get there, or recipes to get these different things? Okay, so let's take, for example, like different attempts at making a cookie. You'll see that they're all kind of similar. You see slight variations, like a little more butter, some use a little more flour. Some ovens are a little bit hotter and colder than others, and you end up with different size cookies. They might be a little bit tougher, but in the end, they're still cookies, right? So we're gonna classify them as kind of the same thing. Likewise, if someone didn't have enough butter and they had way too much flour and they decided to broil it, you're probably not gonna get a cookie. Yeah. Likewise, maybe you have the same amount of butter, but still too much flour, the oven's too hot, you're probably not gonna get a cookie. And that's the idea behind trying to cluster these attempts at baking into these clusters, general guidelines, or like recipes towards these states that we're interested in. And that's what a self-organizing map does at a very high level. You're trying to come up with these clusters or these recipes for these different attempts at baking your hypothetical cookie here. So a self-organizing map is this unsupervised machine learning algorithm, and it creates these generalized clusters / representations that are easier to interpret. And each of our inputs maps to one of these clusters. And I want you guys to think about self-organizing maps as a dimensionality reduction technique. So let's walk into our temporal clustering example. Before I showed like stream flow and temperature, this daily basis here where you have stream flow and then like the previous day's stream flow and temperature lagged by a day alongside red tide and then its previous day's red tide. So you can create like these free machine learning people out there, these, I don't know, what do they call it? Geez, I can't even remember the name. In other words, you're basically creating, what are they called again? Do you remember?
They're cookies?
Yeah, sure. Anyways, what you have here are these daily
Paper cubes, basically, these things?
Yeah, I can't remember the name for them, but basically what they are, they have today's time step and the previous day's time step of your data. So, on January 1st, you have today's and yesterday's, and then the 2nd, you'll have, today's and yesterday's stream flow, and likewise with all the variables. And so what you'll end up with are 365 of these data cubes that are a 3x2 structure, right? And so with our self-organizing map, the idea is to come up with these clusters or these representations or recipes of all of our data. You've reduced the dimensionality of your original data set from 365 times 3 times 2 to 4 times 3 times 2. And what that looks like here, okay, so these self-organizing map clusters should represent your daily regime. So with that, embed your magnitude and direction of all your variables. So, and the cool thing about SLMs, again, is that they match each input to one of the clusters. So in other words, let's just take like January 10th as a hypothetical example there. You see the stream flow increasing, temperature is increasing, and you have this decline in red tide. Again, all hypothetical data, it's not real. And likewise, all these are going to get mapped to cluster 1. In other words, cluster 1 is a general recipe for what's going on in those 4 days there. And what that cluster represents is you have this high but increasing stream flow, this high but increasing temperature, and this decline in red tide concentration. Likewise, for the 14th, that one's going to map to cluster 2, where we have this decrease in stream flow, decrease in temperature, and like no red tide. Okay. And again, these clusters have embedded magnitude and direction of the variables. Okay, so just one important thing when you're clustering data using any of these unsupervised machine learning algorithms is the actual number of clusters, because it's important, right? Like if we, in our previous example, if you have 365 days and you have 365 clusters, you're not really reducing the dimensionality. However, if you have too few clusters, you're missing meaningful variation. And there are a few ways to identify the right number of clusters. One is this Kolinsky-Herabasz index, where you're basically mapping the amount of variance, and you try and find this point of diminishing returns. And for our eventual environmental data cube, we picked this 9x9 self-organizing map orientation, which I'll show later. Okay, so let's talk about what's going into our recipe. Things like our flour and baking soda and whatnot. So first off, we're going to constrain the spatial extent to the Charlotte Harbor estuary. There's a high likelihood that these blooms and these different estuaries are somewhat independent, like in Tampa Bay and Sarasota. It's likely that they're driven by local and global factors, like their local watersheds. So we're constrained to Charlotte Harbor down there in Southwest Florida. And this is actually a cool little video of those red tide prediction maps that we're using there. And the white is the land surface with cloud coverage, which we also had. We had to remove certain days where the cloud coverage was extensive, but you can see, like, if you watch it long enough, you can almost like discern these patterns and really intense blooms starting in the estuary and how they grow and retract offshore and back inshore. So,
so that's like 6,000 days of this, right? Because 2002 to 2024, but minus clouds. So it's, your previous example, 365 days in the hypothetical, but actually way more, and and a lot more butter, flour, sugar, salt, a lot more ingredients. Okay.
Yeah. And so this is as David said, this is the 24 however many years of data that we have, these maps from these red tide prediction maps. And so also they're called tensors, the little box I was thinking of before. Sorry, call that. Okay. So in step 2, we want to calculate the daily distribution of red tide classes at 100%, right? So to show what I mean there is that there's this class distribution as this proxy for daily magnitudes and severity of the region. And every column basically is a distribution of the magnitude of the red tides, or the area of the red tides. And so what I mean by that is, like, let's take a day like October 1st of 2018. What I'm saying is that almost 70% of the area was made up by class 5. So you've likely had like a really intense bloom over that spatial extent that we're looking at over Charlotte Harbor. And just as a sanity check, this is that BSI index showing that same bad, intense bloom event. And you can see it kind of retracting and remagnifying in 2017 there. So that's our response variable. That's our pseudo response variable that we'll be classifying in your self-organizing map. So what are our features, or like our ingredients going into this study? So we're going to use our class 2x6 area percentage coverage. We're leaving out class 1, which I will get to in a little bit, why we did that. So we're using a rolling 7-day sum of the Peace and Caloosahatchee rivers, and those are generally where those two rivers are, Peace River, Caloosahatchee River. Fort Myers air temperature and wind. And we're going to be using Gulf of Mexico altimetry center of mass, in other words, like sea surface height. And what that has been used previously for is a proxy for loop current position, that big ocean circulation pattern in the Gulf of Mexico. And we're going to use a 5-week look back period with 1-week variance for all these different ingredients. Okay, we're going to put that in our self-organizing map. So what do we get when we get this 9x9 self-organizing map configuration to get these funny clusters? Okay, so remember, the idea is to end up with these regimes, and these temporal regimes that have a little bit of the history of all of our variables of interest. So in other words, like what's going on a few days or a few weeks prior with our wind or loop current, stream flow, or temperature and our red tide states. That's what we'll be looking for within our self-organizing map. So this is what you get. And I understand this is a lot, but stick with me for a second. So this is our 9x9 self-organizing map configuration of all of our ingredients of interest. Okay, so what does this mean? So first we reduced the dimensionality, right? We had 8,000 days of this data, but we reduced it to 81. So it's a little more interpretable. However, what do each one of these mean? So remember, on the x-axis of each of these, or the y-axis of each of these, you have all of our ingredients, temperature, lat long, the loop current, wind, our two streams that we're looking at of loading, and our red tide classes. And all those variables are standardized right now. So I'm looking at 5 weeks of historical data there. And each one of these is a recipe for the different states or regimes of all these variables combined. Ooh. Sorry, that hypothetical example is that node 72 down there. Okay, oops. And so can we identify higher level groupings that give us more insight into the relationship between bloom dynamics and environmental features? Okay. And what I mean by secondary clustering is, in our hypothetical example before, can we identify like, hey, this one wasn't edible, and then this one was edible, these higher level understanding of our clusters. Okay, and how are we going to do that? To develop these super clusters of our self-organizing map, we're going to use two techniques, a salmon map, and that one's kind of like a PCA, where it identifies like similarities in hyperspace between all these different different clusters, and awards hierarchical clustering. This is a dendrogram showing the similarities between all of our SOM clusters, and I'll show how we actually ended up with those different super clustering things shortly. Okay, so the idea is to find out these representative states within our SOM. This is just like a little snippet from our 81 or 9x9 self-organizing map configuration. And those 5 clusters are actually represented by those 5 numbers on the x-axis there in the dendrogram. So how did we come up with that? How did we draw that line to come up with those higher level clustering events? Okay, so remember before, now that we've reduced the dimensionality of our original data set, we can actually plot that in the time series, right? So in other words, like node 0 or cluster 0 literally represents days within our time series. That's down there. When we have these superclusters, it helps to make it even more interpretable. And now what we're going to look for is these red tide states that we had mentioned before, like initiation, no bloom, and termination. But okay, so why did we pick 14 superclusters? So we saved this class 1% area to use as a pseudo response variable. So days closer to 100% coverage of class 1 likely have no bloom. So what I mean by that is like, if you have 100% coverage of class 1, it means you have 100% coverage of like no bloom. I understand the axis is a little bit confusing, so that what means, and then 0% of bloom class 1 likely means that you have a widespread bloom. In other words, like most of the coverage is made up by other classes. Okay, so remember, we're looking for, in this first part of this analysis, we're looking for 3 bloom states of interest. That's just kind of like a zoomed in area of this. This termination, no bloom, and bloom initiation. And we're looking for it in this region because, it's highly likely that there's some, when you go from some bloom coverage to no bloom coverage, we're going to classify this as termination. When it stays close to 100% of class 1, we're going to consider that no bloom. And when you start to retract from that or go to get closer to 0% coverage, we're going to call that bloom initiation. Okay. So using our self-organizing map and our wards hierarchical clustering, we wanted to pick a cutoff where we could identify those 3 states within our self-organizing map. And by choosing that particular level, we were able to identify particular clusters within our self-organizing map that gave us this. And this was the superclusters transposed or overlaid of our class one bloom coverage, which gave us these particular cutoff points over time. And so by making that cutoff point and our warrants clustering, we were able to identify days that I could reclassify as termination, no bloom, or additional conditions for bloom, and this bloom initiation phase. Okay, so after we came up with these superclusters, how do we characterize and interpret these superclusters? We did that. Within our self-organizing map, remember, we were now able to get these regions here, 1, 2, and 3 of these SOM clusters that were more similar to one another, that we're calling these 3 different classes. Okay. So what do they look like when we average over all those SOM clusters? In other words, like, if we identify that all these are very similar, when we average over all these standardized values and regimes, what do those superclusters look like? So those that I labeled as termination, this is what those look like. No bloom, that's what that one looked like. And again, these are all standardized values. And this is initiation. Very hard to interpret right now, they're still standardized. So when we unstandardize those values or those aggregates of these 3 phases we're looking at, we end up with these heat maps here. Okay, so supercluster 3 was that those days where I labeled as termination of these blooms. And you can see it, right? So these are all percent coverage. So in every single one of these columns, they should eventually add to 100%. So class 1 was left out. So in this case, it would be like 95% coverage in class 1. However, you can see in all of these that it's decreasing and eventually going to 0. Likewise, in cluster 2, they're all very close to 0 or no bloom. In supercluster 1, you can see the bloom starting to increase again. So now let's actually look at the features associated with these different bloom states. Okay, so our temperature, lat long, wind, and our 2 streams of interest, these are what those unstandardized aggregated values actually look like. These regimes of our cluster data that represent these different bloom states that we're interested in. And one thing that we know to start off the bat is that these 3 states are probably very temperature dependent, they're very seasonal. So we know that termination tends to associate with like colder times of year. These are all in Celsius, and that the initiation phases tend to occur when water temperature increases. So I kind of saved some of the analysis for the end, but this is those aggregated regimes of all of our variables of interest. Okay, so that was initiation. What you can also do with these clusters are look at what actually causes these different magnification events, or what causes bloom growth. We talked about before how there's a lot of different recipes to get there. There's probably a lot of different ways to end up with these really severe bloom events. And so there's 2 states that I've identified here. And we're going to call them supercluster 7 and supercluster 10. And we actually identified those using the same threshold dictated by that Warren's hierarchical clustering. And what those look like, those periods of bloom growth, shown there in the dendrogram, those are the 2 superclusters. So supercluster 7, supercluster 10, again, standardized. And what those look like unstandardized. So we can see the classes or our bloom, our bad bloom classes magnifying in each one of these. Or in supercluster 7, you can start seeing class 2 and class 5 increasing rapidly, and likewise with supercluster 10, where you can see that class 2 and class 5 are getting close to 20% of the coverage of Charlotte Harbor in those particular times. And these are our features of interest and the particular regimes. Okay. So what are our findings and conclusions from this, from all these different superclusters that we're looking at, right? Bloom termination, no bloom, and initiation conditions likely occur every year. They're likely very seasonally dependent. However, these psalms help to identify various bloom dynamics or these recipes of interest. There's these different sequences of events that can drive different intense blooms, like those supercluster 7 and 10 that we looked at previously. However, the main takeaway from these is that this methodology can help to identify common factors among bloom states of interest, whether it be the termination, no bloom, initiation, or these different recipes to get to these bad or magnifying bloom states. So those regimes I had shown previously can be used to characterize the magnitude and direction of these features and how they relate to bloom states. So some of the things that we'll be doing going forward are fine-tuning the feature engineering and the super clustering of these aggregated psalms and creating these finer, coarser temporal resolutions, which you can actually change your self-organizing. Okay, so what can we actually do with this information? So S79, which is where we get our streamflow data from the Caloosahatchee River, that is one of the variables that we have control over because the discharge from that is controlled by the Army Corps of Engineers. So this is actually where we took out. So this is that 9x9 psalm that we had, but we just extracted the Caloosahatchee River, right? So if we know that there are certain regimes that we are headed into, we'll have an idea of what direction and magnitude the Caloosahatchee River may move in to end up with some of our cookies or some of our bloom states that we may be interested in. So the next steps is to use these recipes to identify likely next bloom phases. I'd shown all these different aggregated super clusters from our self-organizing map, but the idea is that, if we know that temperature is high and decreasing, and we know the latitude of the loop current is retracted and the loop, and to the west, and that you have 5 previous days of onshore wind. If we have that information and we know that there is a corresponding streamflow load of the Caloosahatchee River that corresponds to a bad bloom, can we pull certain levers to help mitigate these bloom events? So in other words, like if we know that a really hot oven, in this case, is going to produce not a cookie, can we turn down the oven temperature so that we end up with, not a rock, or end up with a cookie instead? And the other cool thing is that we can apply this methodology to some of the other estuaries of interest, like Tampa Bay or Sarasota. So thank you guys for listening, and I'd be happy to take any questions.
All right, thank you, Nick. We'll take questions from the room and see if we get any from YouTube. We actually had a dozen people on YouTube as well, so. I'm gonna ask the first question, because I didn't get to come to the walkthrough yesterday, so. All right, all the states and all the super states and so, these recipes, can you like give us the highest level summary of like, what is the recipe that you saw for those 3, like the termination, no bloom and initiation? You showed that you had them, but then we didn't spend a lot of time saying what they were. And so that's hard for, we'll keep working on it until you graduate, but for everyone else, what were those recipes? How much flour for no bloom, et cetera? Yeah.
I took it out last minute, but those 3 states were very highly temperature dependent. So when you superimpose those superclusters on the temperature map, which I wish I had kept in, they almost perfectly align with temperature decreasing into the winter time. It's slowly increasing once you get into spring and summer, and always having those conditions that correspond to those 3 states. It seemed like every single year, and things like the bloom severity index show this as well, where there's some background K brevis sampling or concentration within any of the estuaries along the Florida Gulf Coast. And I think those are very cyclical and seasonal. and depend high or just almost all of those things. I think the big takeaway from this one is that there were a lot of different states that correspond to a bloom magnification. And looking for one particular recipe to get to red tide is not really a feasible thing. Because like there's so many, like, we've looked at a lot of different studies where it's like, okay, we have really bad loading one year from the Coosatcha River and it corresponds to really bad bloom magnification. But then other years we have the exact same conditions with no bloom.
Yeah, we had a very intense winter bloom just now that we're coming off of, right? So
Right.
in that case, it's a exception to the temperature rule.
So it's like this, what this one has done is it's helped to identify, because I don't want, the self-organizing map is not a predictive model, but it's a clustering. And so it's not, it literally is looking for historical regime, like reoccurring regimes that have happened. And the idea is that it gives us this roadmap to say like, okay, we are in supercluster 11 right now. These are the particular conditions that are currently going on. In this case, back in 2011, when we got tons of discharge from the Coosatcha River, we seem to get this really bad magnification of growth and blooms. And so the whole idea is
And
to
the supercluster is multivariable, and so it's not like it's, hey, it's summer and there's a lot of, it's summer and the loop current and the wind and the da. And we're in whatever coverage 2 of red tide, and so we can, because that's sort of like that's like the seed,
Yes.
because you're not gonna probably exacerbate from nothing to extreme bloom. You need to have something to be fueling, right?
Right. And so I guess that's the other thing too, where the initial, what we have to take one step back to the red tide prediction maps. As it works both ways. It's probably gonna pick up background red tide that grab samples don't pick up, and grab samples are likely to, be more accurate with the red tide conditions in the Gulf at particular times. So, we have these anomalous red tide events that occur in winter some years, but we don't know if they're actually anomalous. We don't know. There might be background K-brevis cell counts somewhere, whether it be a little further offshore or hidden in, certain parts of the estuaries of Tampa Bay or Sarasota.
Well, so that's good. We actually, following on that same thought about offshore. So Lisa Krimsky asks, she says, fantastic presentation. The question is one of semantics. We know that red tide initiation occurs offshore along the West Florida shelf. Since your models are based on the conditions near shore, technically, aren't you predicting those variables that influence the bloom maintenance stage? And that's an important distinction for management.
It's a great question. And I don't know if that kind of goes one step back to the red tide prediction maps, right? Because they're almost as accurate as the grab samples. They're calibrated on the grab samples and samples that the FWC collects. And so those are kind of limited to that, whatever, like 8 to 10 kilometer offshore range. And we are relying on how accurate those are able to pick up those offshore concentrations, those offshore blooms and how they migrate inwards. But the whole idea of including things like ocean circulation patterns and offshore winds is hopefully identifying the correspondence between these global factors and the offshore conditions to these bloom states. And the other thing that I'm trying to work on now is that you can go back to how these superclusters and SOMs, the days that it's selected for each one of those clusters, and we can actually aggregate the initial maps. And I wish I'd explained this a little more previously too, but why I wanted to break them up into different classes was that I think they're different classes correspond to the offshore inshore component. So what I found is that I think some of the lower bloom classes, I think correspond more to offshore blooms versus some of the really intense blooms tend to be more inshore. So if we know that like bloom class 2 is really intensifying, can I link that to, okay, it's starting inshore and then when you see it, bloom class 6 really magnifying, does it go offshore to inshore in that way? So there's still little bits of that, whether I actually use the original maps or classify the classes as, offshore, inshore, how to discern,
Yeah, and I think, and some of it is just about language, about initiation, and initiation somewhat means like source and cause, and so initiation being a phenomenon that is it's brought inshore, right? And then but you can measure zero concentration, and then you get initiation. You actually cut your maps at 5 or 10 kilometers off.
10 kilometers,
Yeah. So,
10, 15.
yeah, Mark.
Great talk. I mean, and your analogies are great to try to put all that information into context. Two questions. The first is, because temperature seems to be a sensitivity or likely relationship, where's that temperature data coming from? And the second one is more this spatial question where you see the bloom in different places. So do you end up perhaps blurring the response variable, and would it be better to try to reduce that focus, like just inside the estuary versus someplace along the outside, so that maybe correlations between certain driver variables and response variables are more spatially overlapping? No?
So the first one, we use the air temperature. It's just the only continuous variable that we have. We don't have a daily water temperature for that region, but we use the air temperature over 4 meters. So it's close to that constrained spatial extent. And it's a decent proxy for the actual water temperature, but I wish I had left that image in there. But when I superimpose the superclusters onto air temperature, you can almost see, like, as soon as the air temperature as soon as it's low and bottoms out, you end up getting a little bit of a lagged effect of termination. There's termination sun clusters. And likewise, once it starts to increase, then you start to see the superclusters 2 and 1, where they have no balloons emission classes start to increase. So the second one about the maps themselves, the video I showed, that was the spatial extent that we used to get the class area. We just kept it to the shallow water. So, a lot of this comes down to dimensionality reduction, because you're dealing with like the whole coast and days and pixels and all these different variables. And so one way we try to reduce the dimensionality is to just constrain it over shallow. Like, I've thought about even using individual pixels, and then like maybe one in the estuary, there's like one offshore, just to say, like, okay, so does the one in the estuary correspond to Caloosahatchee River discharge versus the one offshore? Does that correspond more to loop current wind and more of these global factors? So it does make a big difference, to answer your question, like how much because if I kept it to the whole coast of Florida, including Tampa Bay and Sarasota, you're losing that direction to that's an important question to ask too.
Any other questions from them? Yeah.
Here's a question, definitely not about the study, just like kind of more trying to understand. So it's like ingredients. Good day. Are we able to access those variables like on a day-to-day basis, like are local municipalities able to like take this research and use it for like better prediction of closing the beach down this day? Like nobody's able to.
It's a great question. Yeah. So the variables that went into this is more of a reanalysis thing. So we looked at variables that we have daily access to, whether it be temperature or this reanalysis of loop current position or all the stream flow data. But in prediction mode, like you're talking about, it's likely that we don't have access to position with the loop current, or luckily things like temperature are a little bit more easy to predict, but some of them, like Caloosahatchee River discharge, is difficult to predict because that's managed and we don't necessarily know. We've had a lot of issues with that in the past as far as knowing when they're going to release a lot of water. So this one was trying to look at these different recipe or baking attempts, right? Try to look at like all the previous historical data to do this analysis, but in predictive mode, we would have to do this in a way where we're just looking at ones that we have daily resolution for.
Yeah, and even so, I mean, like you can't intervene in the loop current position. You can't intervene in the air temperature, but you can intervene in there's one river that you have some control over in your application case. Yeah, the Peace River doesn't have structures on it, right? That you control on a week or a 5-week basis, right? But the Caloosahatchee does. But it's maybe, you could probably predict most of those things. I mean, if loop current position, how rapidly does it change? If you're in a state of retraction of the loop current, right? Right, and you were in, you could have a prediction for the Peace and you can have a prescription for the Caloosahatchee from the Omnicore's 2-week decision making or 1 week or 2-week decision making. The temperature is easy to predict, the winds pretty bad to predict, but I mean, you could have, it just depends on how important they are in getting you from one supercluster to the other. Any more questions from YouTube out there?
Oh, what do you think are gonna be some challenges whenever you try to apply this to different estuaries? There's some different characteristics that you're aware of, at least to this one that you studied versus other ones or vice versa.
It really comes down to how much data is available. So Ron's maps that he created are for the west coast of Florida, so they extend up through like the Pensacola region all the way down, because it's this combination of satellite imagery and the in situ samples. But it comes down to, you know, does the USGS have stream flow gauges for the rivers that we, like the eutrophic rivers in that region? But luckily, a lot of these we kind of view as global factors. So the loop current I view as like this global factor for red tide over the entire West Coast. So it's, and then things like wind, I used ML for this. I could just, and the pixels close to the Charlotte Harbor region, I could, in theory, just do the same thing for Tampa Bay. Temperature, just use like the air temperature gauge close to the Tampa International Airport versus Charlotte Harbor. So it just becomes like a data. It's available.
All right, let's give another round of applause to Nick. Thank you. So next week, we have another in-person presentation from Dr. Beatriz Marin-Diaz, another postdoc researcher in the Center for Coastal Solutions, and her talk is bamboo amendments to prevent seagrass loss.
Next week.
That's right, 2 weeks. Yes, that's why you're making that face, right? Yeah. So yeah, spring break is next week. Enjoy. Please come back with a tale or two from somewhere beautiful, maybe a wetland or at least a watershed. Okay, thanks everyone. See you next time.